Hey guys, welcome back. Today we're going to go ahead and cover some ground in Chapter 13. Last time we talked about Punnett squares and some simple inheritance, and today we're going to take a look at how Mendel kind of came up with these ideas. And so this is what your last slide looked like. We just talked about P, F1, F2 for generations. So here there's a couple possible events when we're talking about monohybrid crosses. If we have a big P allele from sperm and a big P allele from egg, then the zygote that's made is big P, carries both alleles for big P. Other ways we could do this: sperm big P, egg little p, zygote that they make is big P, little p. Opposite of that, sperm is little p, potentially egg is big P, zygote they make, big P, little p. By convention, you write the capital letter first, even though that's not the order of this table here. And then, of course, finally, the sperm, little p, egg, little p, zygote that is made is little p, little p. So that's the idea that we understand now for simple Mendelian genetics, and I'll show you how he got there. So Mendel used these monohybrid crosses to find our dominance relationships among the pea plants. And when he crossed plants that were true breeders for white flowers with plants that bred true for purple flowers, all the F1 plants had purple flowers. So he took all these plants that were just purple and all these plants that were just white, and he crossed them and planted the seeds, and all the seeds were purple. And Mendel thought, I don't think the white is necessarily gone. I think the purple is in the way of the white, basically. He didn't think the stuff that made flowers white was completely gone. He thought it was hidden. And at this time there was no, DNA, genes, alleles, none of that was around. He just thought whatever made flowers white was still in there somewhere. And so then what he did was he took one of those purple plants and he crossed them with the white plant. And he got this, where they're all purple. And they're all, he still thinks there's white in there somewheres. And so what he decides to do, which is really smart, is he takes two of these F1 generations and he crosses those to make an F2. And so they segregate big P, little p for one parent, and then big P, little p for another parent. And he dries out these seed pods, he plants those peas, and they come up, and most of them are purple. And at this point, some of them are white. And so he does this with a lot of flowers. And it comes out to a 3-to-1 ratio. 75% of these flowers he plants from this cross come out purple. 25% come out white. And so now he's shown experimentally the stuff that makes flowers white is some sort of transmittable information to kids and grandkids that can be hidden by another trait, and it can show up later, right? And so that was kind of a cool example of how he figured this out. So like I said before, he observed a phenotype ratio of 3-to-1 in his F2 crosses. If I lost you on that diagram, I can show you in the classroom. We can work through all these Punnett squares together. Just ask me, and that'll be easy peasy. It matched the probability of the little p genotype in the offspring of a heterozygous cross, and that was the basis for Mendel's law of segregation, which is these cells, diploid cells that have a pair of genes on pairs of homologous chromosomes. This is today's language. And two of these genes of each pair will segregate during meiosis and end up in different gametes. And so what that basically means is this right here. Big P goes to one cell, little p goes to one cell. And they are separated into, since it's plants, different pollen cells. Big P goes one way, little p goes one way. Or if it were little p goes one way, the other little p goes the other way. They go into different cells. That's segregation. Segregation means to separate. His law of segregation was that the alleles, or rather, maybe I shouldn't say alleles, the genes, sure, the gene pairs separate during meiosis. That's the law. So the other law he had was independent assortment. What he got when he did these tests were that it was kind of random as far as what plant or what pea seed was actually going to be the white flower. It wasn't like always the second seed pod down the husk of the pod of peas, or the third or the fourth. It was pretty random. And so he did these dihybrid crosses to test for dominance relationships between alleles at two loci, two different locations. And of course he didn't know alleles were at two different locations. This is all updated. And so we do two traits. Individuals that breed true for two different traits are crossed. Big P, big P, big T, big T. So I think it's purple tall. Little p, little p, little t, white, short. And that's how tall the plant grows. And so during meiosis, the members of a pair of genes on homologous chromosomes get distributed into gametes independently of other gene pairs. So what that means is this. If we start to split this cell into meiosis, and this P here goes into this cell over here, that has no effect at all on these two. Now, had Mendel had the misfortune of working with genes that were linked, his data would have been crazy and would have made no sense. But if genes are not linked together, they are not inherited together. And so Big P can go wherever he wants. That does not affect where T goes. This guy could go this way. He could go this way. It doesn't matter. They segregate independently of other alleles. That's what that means. So random, truly, truly random. And so here we have the dihybrid P test, a tall purple flower, a short white flower, and then we get the parents, which are heterozygous. I'm sorry, these are the parents. We get the children, F1, heterozygous for both traits. That can make 4 types of gametes. It can make a big P and a big T, because it's random. Could make a big P and a little t. Could be a little p and a big T. Could be a little p and a little t. Hopefully I said all those right. So you just start to do your arrows to figure out what alleles you could possibly make. And that's how you do that. Get you 4 different combinations. And so now this part's tricky. Put a gamete instead of the 4x4 block, or a 2x2 block, we do a 4x4 block. So here's your big P, big T, big P, little t, and we're doing them the same exact way here. Little p, big T, little p, little t. And so now Mendel's math skills are going to come in handy because he's going to plant a bunch of these seeds and it's going to be crazy. This one here gets big P, big P, big T, tall, purple flowers. Okay? This guy way down here, little p, little t, short, white flowers. Short, or rather white, tall flowers. Purple, tall flowers. Purple, short flowers. And so he starts to get these ratios. And this works out into a 9 to 3 to 3 to 1 right there. You're going to get 9 phenotypic ratios of the dominant traits, 3 heterozygous dominant for one trait, 3 heterozygous dominant for the other trait, and 1 recessive for both. That's what that's going to look like. And so that's a dihybrid cross. We'll practice that as well. And so then here's your independent assortment. Your F2 phenotype is 9 to 3 to 3 to 1 because there's 4 different phenotypes. Individually, each dominant trait has a 3 to 1 ratio, but when you combine 2 of those together, it's 9 to 3 to 3 to 1. The inheritance of one trait does not affect the inheritance of the other. Sometimes. Most of the time. In pea plants, we got really lucky. That was not the case. In humans, that can be kind of tricky. But yeah, generally, if as long as a gene is far enough away from another gene, its inheritance does not affect the other. So that's very cool. That's the law of independent assortment. All right, so let's talk crossovers. In meiosis, you guys know that crossovers occur. We talked about that, I think, last chapter, that we talked about the great benefit of genetic diversity that crossovers give us. So independent assortment also occurs when our genes are on the same chromosome, but they have to be far enough apart that crossing over occurs between them frequently. Because if crossing over occurs frequently, we're still going to get random distribution to the different sides of the cell. All genes on one chromosome are called a linkage group, because unless that chromosome is crossed over, that chromosome will be pulled away in meiosis, and all those genes will be inherited together. Those are linked genes. And so you may very well have, 2 genes in a human, or 3 or 4 that are linked genes, and they might not give us that perfect random distribution. These genes on the chromosomes of the pea plants were far, far away, so crossing over made them essentially random. So we got really, really fortunate for that. Genes that have loci very close to one another tend to be inherited together during meiosis. Like I just told you, they're so close together that when they get drug apart, when that chromosome gets drugged, they're going to be together, these 2 genes. And if they're super close together, the odds of crossing over occurring in a space in between those 2 genes is very small. Does that kind of make sense? I hope it does, because I'm just talking to a computer here, and I can't see the confused look on your faces. I hope it makes sense. Maybe this really convoluted diagram will help. Sorry, that's rude. I shouldn't be so rude. Here you go. 2 homologous chromosomes. This is a reproductive cell. They could have been split up like this, or like this. Either path here could happen during meiosis. Pardon me. We split them up, and you can see a little bit of crossing over has occurred. We split them up, meiosis 1. A little bit of crossing over has occurred here as well. We split them up in meiosis 2, and we have one copy, a little bit of crossing over in there, one copy. But all these combinations are different. And so really that's the important thing. Independent assortment, who's to say where that guy is going to end up? I don't honestly know. We got all these combinations. He could be here, or here, or here, or here. Not there. He's not there or there. But no, you never know. He could have ended up over here. That's the independent assortment. I don't know where that little chromosome right there. I don't know where he's going. Could be anywhere. Could be 4 different cells. It's random. So here's linkage groups. The farther apart 2 genes are in a chromosome, the more often crossing over occurs between them. You think crossing over in a chromosome, the more space you have between your neighbor, the more likely that chromosome is going to be able to cross over in between you guys. Linked genes are very close together, and crossing over occurs between them very, very, very rarely. That's why we call them linked, because we see them inherited together almost all of the time. And so the probability that a crossover will separate alleles of 2 genes is proportional to the distance between those genes. More distance equals higher probability of crossing over. Make sense? Less distance equals lower probability of crossing over. And if 2 genes are right next to each other, they're pretty much linked. That's what that means. So let's stop there, because 13.5 is beyond simple dominance, which is where we take everything we just learned and just shatter it. Not really. It's not really that bad, but it does get a little more complicated. So we'll stop there, because you got a lot to chew on and think about. So thanks for your time.